So now that you became geneticist, let's go back to the report as you receive it. And if you take all the information that I provided you with, you can now read it very differently. So the first information here, which is provided at the top, tells you what has been done to produce the report. And it says whole exome sequencing. And now that you know what the exons are, which are the really critical part of the genome that will be translated into the protein, you know that in that case the hospital has done is that they read only the exons, not the introns or not the promoter or not other genes, but only the exons of a given panel of genes. And why am I mentioning this? This is because most of the mutations in the FOXG1 gene are indeed in the exon, right? So it does make sense to do exome sequencing. But this is not always the case. And I came across one family who contacted me. They did first exome sequencing and nothing came out. No one knew what the kid had until they did another type of analysis, which is called the whole genome sequencing. So here, instead of focusing only on the exon, which is the case of exome sequencing, what the clinicians did is reading the entire gene, the entire FOXG1 gene. And it turned out that there was a mutation in here, which is in what I called previously the promoter, which are regulatory regions. And that has as well consequences. To go back to your question, even mutations sometimes in introns can have consequences. But to detect that, you need to read the entire gene, actually the entire genome. And that is why now the new technology allows to read everyone's genome entirely to cover absolutely, as I was mentioning at the beginning, the 6 billion letters. And that's how we get better and better at providing people with an accurate report.
Okay, so some families who got whole exome sequencing and may have an undiagnosed condition for their bub, but their bub's not progressing usual, in a normal fashion, they may want to go get whole genome sequencing. I imagine that's largely available these days.
So I believe there is an initiative, at least in America, where I can't remember exactly the name of this initiative, but the idea is to indeed take all those families that are affected but undiagnosed, where the diagnostic is not given because we don't know what's happening, and go through analysis such as the whole genome sequencing and try and find out with the latest technology. There is actually now the third generation of sequencing sequencer, so the machines that are used to read the genome, that allows to go even deeper. And we hope that this new technology will indeed provide families with a diagnostic. Yes, absolutely.
Just one more question. Is it possible to have a FOXG1 mutation in an exon and then also, for example, in a promoter?
Yes. So
could you find it in multiple spots? Yeah.
Yes. So most of the mutations are what we call discrete mutations. It's one letter, but they are much bigger mutations. Sometimes the entire gene is, for instance, reverted to what we call translocation, where it's exchanged with another gene on another chromosome. So the mutations can be small, big, unique, or multiple. So indeed, you can have multiple mutations in the FOXG1 gene. Yes, absolutely. But now that you really have a much better understanding of the genetics, I just want to guide you through this, which is really important, and that is included in every single report. If you read with me here, the patient is heterozygote for the FOXG1 gene, and then it says C584GCP ARCH195 Pro. So obviously, a family provided with that without any genetic counseling or any explanation would have no idea what it is. However, now that I gave you the information, you will immediately understand that the C here stands for coding. So we're talking about that at the genomic level. The mutation is at the position 584. The nature of the mutation is this G letter has been replaced with a C. And the consequences at the protein level, which is why you got this P here, is that the arginine, the amino acid arginine at the position 195 has been changed with a proline. So if you go back to the code that I showed you just before, you will indeed see that this mutation induces these changes at the protein level. So that is really the core information that is provided to the families, and that unfortunately most of the geneticists provide without any explanation. So I hope really tonight the idea is to try and decode this report, and that is now sorted out. But I'm gonna focus on 2 or 3 more quickly pieces of information that are provided here, and that are not obvious for the families. So the first one is, as I read a few seconds ago, the patient is heterozygous, and this is repeated here. Het means heterozygous, and this AD here means autosomal dominant. Don't worry about dominant. I will introduce you to the 2 different concepts of heterozygosity and the concept of autosomal. So for that, I went through quite deep into the genetics, and I told you, provided you with plenty of information. I told you, for instance, that you have in each and every single of your cells 6 billions of the letters, and some of them making up genes, in that case, the FOXG1 gene. But what I didn't tell you until now is that for each gene, you actually human beings have 2 copies of every gene. And I mentioned a bit before that it was necessary because having 2 different copies allows you to have 2 slightly different copies. And for instance, you can see that on that schematic here, that this is what allows the genetic diversity. This is what makes you, Steve, you, and me, as in I have blue eyes, you may have brown eyes. So it is really important to understand that you don't have 1 FOXG1 copy, you have 2 FOXG1 copies in every single cell. And why is it so important? It is because if you look at it here, most of the individuals have 2 identical copies, as it is described here in blue. But unfortunately, if you have one of the copy which carries a mutation, when the parents have this mutant copy, their children can inherit any of these combination here. They can inherit 2 different copies of the mutations. This, in the case of FOXG1, is actually lethal, and only this combination exists in the case of FOXG1. But the concept here that I wanted to introduce is if you have the same copies, it doesn't matter whether it's the mutant one or the normal one, but if your 2 copies of FOXG1 are identical, we call this homozygous. However, on the contrary, if your 2 copies are different, we call it heterozygous. And all the FOXG1 children are actually heterozygous. That means they have 1 copy that carries a mutation and 1 that doesn't carry a mutation. And why is it so important? This is because unlike most of the genetic disorders, only one copy carrying a mutation is enough to induce the disorder. In most genetic disorder, you need to have the 2 different copies of your gene that carries mutation to have consequences. And you will hear sometimes that FOXG1 is a case of haploinsufficiency. if you hear that, that also means it's a synonym for this, which means that only one of the two copy, if only one of the two is mutated, carries a mutation, then it's sufficient, unfortunately, to induce the disease, right? So that was the concept of homozygous and heterozygous, which is included in the report here. And the last concept is actually the concept of autosomal. What autosomal means, as opposed to allosomal, is that the FOXG1 gene is actually included into what we call the somatic chromosome. So there's different types of chromosome, which are subsegments of this long stretch of DNA. And some are specific. We call them the sex chromosome. You've heard of the XX for the female and the XY for the males. And FOXG1, the FOXG1 gene is actually not on the sex chromosome, is on the other ones, which are called the autosomes, which is why FOXG1 is said to be autosomal. And why is it so important? I will finish really with that one, is because if you look at the very last piece of information that's provided here, which is called the phenotype. In this report, it says Rett syndrome congenital variant. And that is something, to me, in 2022 shouldn't be on this report. Why? Because unfortunately, at the very beginning, FOXG1 has been inadvertently diagnosed as being Rett syndrome. You know that, Steve. Sometimes, still today, FOXG1 is called atypical Rett syndrome. But the problem comes from the fact that we realize that, so Rett syndrome has been discovered first, right? But we realized years later that FOXG1 was actually a different disorder because it involves a different gene located on.
That was about 15 years ago, right? From today?
Yes, absolutely.
So about, yeah, okay. Yeah.
And so just to show you that, to me, indicating that FOXG1 is a case of Rett syndrome congenital variant is a misinformation. If I go back to the difference between autosomal and allosomal, sorry, is that look at the FOXG1 gene. It's located on chromosome 14 in human, which means both boys and girls can be affected. However, Rett syndrome, as you know, the mutation is actually located in the MECP2 gene, which is on the X chromosome, and therefore only girls are affected. And the problem is, because the kids have very similar symptoms, then for years, FOXG1 syndrome has been simply considered being Rett syndrome, and you still have that on the genetic report. But these are two very different disorders. And the interesting part, though, is that although they are very different, research that focus on one can certainly help on the other. And we often talk about FOXG1 as helping, the research on FOXG1 helping research on other genetic disorder, and this is actually very important.
Fabien, I'll just jump in there for a second, say that I think it's important to set the scene for the families, is that, for example, if we were lucky enough to find a complete therapy and a cure, it wouldn't necessarily mean that Rett or Fragile X or any other condition we're able to find it, because the genetics are just so different. And I think from time to time it can be easy to get excited and say, well, that, you know, there's some therapy or there's a cure for this. Why can't it happen for us, even though the children and the people present quite similarly? I just think it's worth setting that stage, for realistic expectations.
That is probably,
I imagine you would agree.
I fully agree, and that is probably why I wanted to really pinpoint the fact that even on a genetic report, the information can be somehow not accurate. And the fact that it's still, FOXG1 is still called Rett syndrome, it's actually still the case, by the way, in scientific publications as well. is something that needs to stop. And we need to consider the two different diseases as separate. And still, as I said, research on one will probably help the other, but that doesn't mean that if you cure one, certainly you won't systematically and automatically cure the other, because they are actually different, as I showed you at the genetic level, even if they look very similar in terms of symptoms. So this is really, really important, yes.
I think you're just coming up on the last point that I was going to make, which is about the mosaicism.
Yes, so absolutely. So that brings me to the very last information that is provided here on this report. You can read the patient's mother is mosaic for the variant. The concept of mosaicism is one that I will also illustrate here. When fertilization occurs, the first cell that makes up the embryo is called a zygote, and this first cell will divide and become two will become four, and then eventually you will become an entire individual with millions of cells. And as I told you at the very beginning, every single cell within your body, I said that all these cells carry the exact same genome, the exact same sequence in your DNA. In fact, this is not systematically the case. Sometimes, after few divisions, one of the cells will carry a mutation. A mutation will appear in one of these cells, and then, as you can see here, when the individual is somehow finalized, not all the cells within your body carry the same genetic sequence. And why is it? This is what we call a mosaic individual. Research has shown that the mosaicism within a population is really under-evaluated because we never look at it. Most of the time, we don't even know that we are mosaic. But why is it so important in the case of this report here? Is because it helps. This sort of information is critical for people like us, for researchers that study FOXG1 syndrome. Because let's go back to the very assumption that your mutation induce changes into the FOXG1 protein. The FOXG1 protein is not functional anymore. What everyone hopes is that if you restore this FOXG1 protein to what it was originally, then somehow we will cure FOXG1. But the thing is, no one knows to what level we need to bring back the FOXG1 to what it's supposed to be. Is it 100%? No one knows. But this, for instance, can give you an indication, because here it says that the mother of the patient had 5% of their cells that has the mutation. So the mother doesn't have FOXG1 syndrome, yet 5% of the mother's cells carry the mutation, right? So maybe if we don't bring back the level to 100%, but to 95%, we're still going to be able to somehow reverse completely the course of the disease. So these are very important indications that you find on the report that may not be understandable for the family, unfortunately, and I advise them to seek advice. It's sometimes difficult to find someone who has time and the expertise to explain that. But I also really invite the families, as much as they can, to share their reports, because these indications are critical for us researchers, and that's where the exchange between the families and the researcher is really critical. And
I mean, just quickly on that point, sorry, before you go there, I know that we know of another family who has a FOXG1 child, and that child is mosaic for that, and the mother had 10% affected cells. So I think we know where we think we can go back closer to 90 and have a healthy, a healthier person.
That's correct. I've been in touch with this family, and indeed, the mother has 10% of the cells. Now, we hope that 90% non-mutated FOXG1 would be enough, but we don't know. Yet we have some indication, so it is critical, but it still needs to be tested. Now, let me go back to what the family can do to help us. Is one of the very rare yet great initiative that has been taken by the FRF is this registry, as it's displayed here, where the family can freely and should provide genetic information, including genetic reports, into a database, into a registry. And as much as maybe the family don't realize the impact that it has for us, as I try to explain going through this, the bits and pieces of genetics, this, for someone who was interested in this and whose job is like mine, this is critical and valuable information that we need to have to be able to help the family in return. So I wanted really to bring that to kind of encourage the families to bring samples and genetic report into the FOXG1 Research Registry, which is a great initiative.
I just add on to that, Fabien, that from a foundation perspective, we would really, really appreciate that, as well as skin samples for the lab, and participating in a natural history study is also really, really important for that, significant data that we can get to folks like yourself. The more data you have, the better research is going to be. So I just encourage the parents and the family to really consider that if it was something that they were able to do.
Absolutely. So I'm really finished with kind of the take-home message that I wanted to bring to the families tonight. Hopefully it wasn't too complex, because it's recorded. Everyone will have the ability to watch it at their pace. Just to let everyone know, there is already a presentation on YouTube, another one that is done by, I believe, a father. I watched it. I think it's a good idea to watch the one we're doing now and then watch this one, because you'll be provided with the basic of genetics that will help you understand once again this video done by last year by a father.
That was done by Ryan, who is our CFO in the States, and it's a very, very good one.
Mm-hmm.
What's interesting from our discussion and that video is that you'll also see a difference in the way the genetic reports are presented, because there's no one standard report. They all carry the important information, but it's displayed a bit differently. So you might go through this video or that video and just observe differences. That
is correct. Having said that, the very core information, I've seen reports in 4 different languages so far, French, English, Spanish, and German. And despite being done in different countries and in different languages, the very core information that I provided you with, the code between the genome and the protein, the type of mutation, etc., it's almost systematically included in the report. But yes, you're right, there are subtle differences, so the more the merrier in terms of being provided with information.